Welcome. My name is Chris Baker, and this is my colleague, Milo Johnson. And we're both graduate students at Harvard, and we work in Michael Desai's lab across the river on evolutionary biology and evolutionary genetics. But today we want to talk to you about rogue genomes, evolving and engineering with selfish genes. To start, a little bit of exposition to make sure we're just on the same page. What is our genome, right? People throw the word around genomics a lot, genomes a lot. And basically when we say this, what we're referring to is to all of the DNA present in each cell of our body. And since the DNA in each cell of our body represents the inherited genetic material from our parents, this essentially amounts to the genetic instruction book for making us and for making us human. And like many other books, our genome has chapters, and we call these chromosomes. They come in pairs, and there are 23 of them. One member of the pair comes from your mom, one member of the pair comes from your dad. And also, like chapters in a book, each chapter, each chromosome is made up of many sentences, or what we call genes. And we'll come back to what a gene is in just a second. And each of these genes, in turn, is made up of a string of letters that we call DNA. And here it is spinning beautifully, the double helix that we're all familiar with. It's like a ladder almost, with each rung on a ladder representing a different letter. And now we're going to talk a lot about DNA and genomes throughout the talk. So I just want to get a couple of things straight right off the bat. So we could draw this on the screen every time we want to talk about DNA. The double helix, it's beautiful and all, but it's also kind of a little too specific and a little too complicated for what we're trying to do. We could write it up as the letters, where each letter, each C, T, A, and G represents a rung on that ladder. But even this is a little too much in the weeds for the concepts we're trying to relay and twist around tonight. And so instead, we're going to just think of it as this monolithic, boring, gray block. That's DNA. It's an oversimplification, but it's a really useful one. And a meaningful part of this block would be, we would call a gene. And it's meaningful because through a number of intermediates, such as RNA, mRNA, etc., it produces proteins. And proteins are super important. They are the molecular machines that basically make every part in of your body work. It's responsible for memory, for seeing, for breathing, for pretty much anything you can think of. Proteins are responsible for that in your body. So kind of to zoom out again, your genome, your instruction book, is essentially what's responsible for making you and for making you human. And I ask you to suspend disbelief with this slide, though, because as we all know, Beyonce is superhuman. But just, use your imagination. And so the next question is, and the one we were able to answer around the year 2000 with the Human Genome Project, is, okay, what's in our genome? What is in our instruction book? And the answer was a little surprising. See that little sliver there in red? Those are the genes that make proteins. And that represents just a little over 1% of your entire genome, all of the DNA in every cell of your body. That's a little weird. So what's this other 98.5%? Well, this section on the left here, on the right here, is other DNA. And we're not going to talk too much about this tonight. There's some interesting stuff in there like introns, which kind of break up genes into different chunks, and some miscellaneous repeats. But really what we're more interested in is this chunk on the left, this 45% that we've called transposons. What the heck are transposons? And this is what we're going to talk about in today's lecture. Not only what are they, but also how do they work? What makes them tick? How are they discovered, and how diverse and abundant are they in the natural world? And then we're going to get into a really important question, I think, that we could talk about for hours on end, as Steph and Katie said. Are these transposons our friends, or are they our enemies? And maybe more specifically, I'm going to take the position, I'm going to argue against Milo on this one, that transposons are ill-mannered, malintended parasites that want nothing but the worst for us. Whereas Milo is going to take this position that not only are they're our friends, but in a really fundamental way, they're actually us. And finally, at the end of the talk, we're going to talk briefly about how scientists have tamed transposons for their own purposes and are now using them as tools to probe basically all these aspects of biology that set the rules of biology and try to understand how life works at its core. And so with that, I'm going to turn over to Milo for the first introduction.
Thanks, Chris. All right, hello everybody. So I'm gonna go ahead and get into this with the question that that Chris asked before. If I can figure out how to use the clicker, maybe. So what is a transposon? Well, a transposon is a piece of DNA that pastes itself in the genome. And in this kind of cartoon view that we're using today, we're going to show transposons as this kind of blue block on the gray DNA of the genome. And we're going to specifically show the edges of the transposon in brown. And this is because the edges or the ends of the transposon happen to be really important for a lot of things that they do. So there are two main types of transposons, and we can think of what they do and what they are based on kind of familiar operations that you might be used to doing in Microsoft Word. So a DNA transposon cuts and pastes. And if we think of this gray block and these letters as a section of the genome, what a DNA transposon is going to do is cut itself out of the genome and paste itself somewhere else in the genome. The other type of transposons are retrotransposons. And again, similar to what you might be used to in Word, a retrotransposon can copy and paste itself somewhere else in the genome. So these are the two types of transposons, and I've kind of given you this broad view of how they work, but how does it actually happen? So for DNA transposons, we can go into a little bit more detail and actually describe how this cutting and pasting happens. So this is specifically about a transposon called TN5, which isn't particularly important, but that's the name of the transposon. And these two gray strips, again, are just the genome, and there's TN5 sitting in the genome, kind of hanging out. And what it's going to do is, through the process Chris talked about earlier, it's going to make a protein called TN5P. And what TN5P will do is it will locate these brown edges of TN5, and it's going to cut it out of the genome and pick it up like a little handbag. And the genome will be repaired there, and TN5P will go cruise to somewhere else in the genome. It could be nearby, it could be a whole nother chromosome. It's going to make a little incision, and it's going to unfurl that transposon into a different place in the genome. So the really important thing to remember here is that the protein TN5P that does all of this is actually created by the transposon, the little blue element. So in this way, it's kind of a self-contained cutting and pasting machine. So for retrotransposons, it's a little more complicated. And tonight, I'm not going to have time to go into all the details of exactly how it works, but you can imagine it being really similar to what I showed on the previous slide with the DNA transposons, except in this case, instead of cutting and pasting, it's going to copy it out of the genome and paste it somewhere else. And again, the important thing to remember is that all of the proteins involved in copying and pasting it will be created by the retrotransposon itself. So if we think about how these transposons live and what they do, I think it's nice to refer back to something you might have heard if you were here last week, which is that for cells, the dream of every cell is to become two cells. And in the same way we can think of transposons, that the dream of every transposon is to become two transposons. So we can think of transposons as these kind of selfish replicators that are going to try over time to go from 1 copy to 2 copies to 4 to 8 and so on. So at this point, if you've been paying attention, you might have a nagging question at the back of your mind, which is that this picture makes a ton of sense for the retrotransposons that copy and paste, right? But for the DNA transposons, where they're just cutting and pasting, if you've ever sat on Word and just cut and paste a word over the document for a long time, you You won't end up with more copies of it. So it's a good question, and there's a few ways that DNA transposons can get around this problem. So I'm going to explain one possible example. So again, if we think about cells dividing, one cell dreams to be two cells, but before it can be two cells, it needs to have two copies of the genome because both of the new cells need a complete genome. So if we think of this gray bar as the genome, before the cell can divide, it needs to split the genome into two identical copies. So this is DNA replication. It's pretty well understood. So one way that DNA transposons can get around this problem of just cutting and pasting and not making more copies of themselves is through this DNA replication process. So here we see the genome, and in it is our friend the transposon. And here we have a snapshot in time where part of the genome has been duplicated. So you see the transposon has already been duplicated, but parts of the genome haven't. So if during this time, the DNA transposon cuts itself out and pastes itself in front of what we call the replication fork, you can kind of see why it might be called a fork, then you'll get this picture. And then you can see that if the duplication finishes, you'll end up with two new copies of the chromosome or the genome, and now one of them actually does have two copies. So it's kind of a sneaky little trick that DNA transposons can do to make more copies of themselves, even though they're just cutting and pasting. And there's a couple other ways they can do this, but this is my favorite example. So this brings me back to this idea that for both DNA transposons and retrotransposons, they're kind of these selfish replicators. And in this way, transposons are sort of alive. And I don't think any scientists out there, very few scientists you would meet, would actually call transposons alive. They don't eat, they don't metabolize, they live inside the DNA, they're just kind of molecules, right? But in this specific way, they are really similar to life. So all of life, in a sense, is here because it replicates. And transposons are the same in this way, and because of that, we can kind of think of them as kind of being alive, and we can certainly think of them as evolving. So just to drive home this point that all of life are replicators, I want to take a quick poll. So raise your hand if you have kids, if you have children. Okay, there's a few. All right, raise your hand if your parents have children. All right, that's almost everyone. Right? That's pretty good. So in a sense, like if we think about what is around today and what is living, everything that's around today and everything that's living is here because it replicates, right? And so this is the way in which transposons are kind of alive and transposons are kind of evolving. Definitely evolving, kind of alive. So just to wrap this section up, I want to make one last point, which is that transposons don't only spread within a genome. They also can spread within a population or a species. So this is a really cool example of a DNA transposon called the P element and a little fly called Drosophila melanogaster, which happens to be one of the best studied animals. It's been studied for over a century. And so because of that, scientists happen to know that there were a few individuals in 1950 that had the P element in their genome in Florida, around Florida, sorry. And over the course of 3 decades, the P element was able to spread through the population so that by 1980, if you grab one of these Drosophila melanogaster from nearly anywhere in the world, it would have at least one P element in its genome. So these transposons have the ability to not only spread within the genome, but to also spread within populations. So in summary, transposons are a major part of our genome. As Chris showed, they take up a huge portion of our DNA. There are 2 types of transposons, and they've evolved sophisticated ways to cut, copy, and paste themselves around the genome. And then finally, we can think of these transposons as these kinds of selfish replicators that can spread within a genome or within a population. And so with that, I can pause to take audience questions if there are any audience questions. Yeah?
Is this what Barbara Mackey, when Doc was working on?
So the question is whether this is what Barbara McClintock was working on. And yeah, you're exactly right. And that's coming up next. A brief description of that story. But yeah, absolutely.
So what are the beneficial characteristics of the transposon?
So the question is, what are the beneficial characteristics of transposons? And that's another question where I'm tempted to say just hold on, because that's almost the topic of the rest of the talk. Is, are they beneficial? Are they not beneficial? What exactly are they doing? So I'd ask you to hold on to that, and at the end of the talk, if you still have questions, which you probably will, we could talk about it more. Yeah.
Have they existed since life existed? Or did they come up like
Yeah, so that's a great question. So the question is, have they existed since life existed, or did they kind of crop up? I think the answer is we don't know, for sure. I would speculate that they haven't existed quite since life existed, but I wouldn't be surprised if they've existed certainly a really, really long time. And the origin of them is a confusing question as well, which is tough to understand. Yeah.
So as they replicate, do they change at all, or are they the same characteristics throughout the whole chain?
Yeah, so the question is, as they replicate, do they change at all, or do they stay the same? And I think in the same way that, like, as the genome replicates, it stays the same, you could say that transposons, when they replicate, they stay the same. So just like we, over the course of time, will acquire mutations in our genome, and those mutations will be passed down to our kids, that same kind of process will happen with transposons. But in general, for one replication, you would expect everything to be copied perfectly. Yeah. Any more questions? Yeah.
Now, does the TN5P look for a specific place to copy itself to, or is it kind of random?
That's a great question. So the question is whether the TN5P looks for a specific place to copy whether these transposon proteins look for a specific place to copy the transposons to, or whether it's random. And that's an active area of research in transposon biology. And for some transposons, I don't actually know for sure for TN5P. For some transposons, we know that it's fairly random. And for some transposons, we happen to know that it's very specific. It goes to a few specific regions or a repeated region in the genome or something like that.
It seems in some way analogous to Cas9, that was wondering if the ones that go to specific regions have something analogous to guide RNA.
Analogous to guide RNA?
To guide specific, to match to specific regions.
Yeah, absolutely. I think there's a lot of parallels there, and Chris knows this better than me, but I think there's even explicit connections between transposons and CRISPR-Cas9.
Right, and just to add on to that, so Cas9, just for everyone to know, is a new technology that people have found in bacteria, and it's a way that we've developed of making really precise edits to pretty much any genome you can think of. So it's a really kind of, it's called CRISPR as well. And it seems that, to answer your question, actually the proteins that are part of Cas9 actually were transposons in their evolutionary history, and they've been called Casposons, and they since have evolved into a more proper Cas9 system in bacteria.
Yeah.
Are transposons then the things that, when in DNA, suddenly some aberration occurs, are they responsible for that?
So the question is, like, when there are suddenly aberrations in DNA, are transposons responsible for this, right?
Yeah, like an eyeball begins to develop or something, no one else has it.
Yeah, so they are one mechanism by which that can happen. So there's other types of mutations that could cause cause weird things. If you think of most mutations aren't going to cause much to happen at all, but certain ones will cause weird aberrations or cause changes, and transposon insertions certainly are one type of mutation that could cause these differences. Yeah.
Last question.
Are there does this constantly happen within the genome, or are there specific time frames within this activity occurring?
So the question is whether this happens constantly or whether there's specific time frames that it occurs. I think again it's different between different transposons. There's a lot of different kinds of transposons. The main example that I keep finding in reading about different transposons is that a lot of times it is happening along with DNA replication, like I showed for the DNA transposon example. And I think even sometimes for retrotransposons, that's also the case, that it will time with DNA replication. There's a whole variety of reasons why that might be the case. But it's one of those things that's known for some transposons and not known for other ones. All right, so with that, I think I'll turn it back over to Chris.
Thanks, Milo. Yeah, and chances are that a lot of your questions that you have now may be answered as we go through the talk, and other ones certainly will come up. And we'll pause again. All right. So next we're going to talk a little bit about how these were discovered and how transposons exist in the natural world and in our genomes. So the story really starts with, as this gentleman mentioned, Barbara McClintock. And she was a crop geneticist starting around the 1940s. And kind of in hindsight, that was a tough place to be for her for two reasons. One, being a female scientist in the field of crop geneticist, which was very male-dominated, was simply not a cakewalk for her. I don't know all of the stories of the hardship she felt through that, but there were many. And secondly, being a geneticist in the 1940s was very difficult because it was not until the 1950s that not only was the structure of DNA not discovered by Watson and Crick and how it's replicated and how it's regulated and all of these important things, but they didn't even know that DNA was the material that was inherited and responsible for being the instruction book of the cells. So there was very little to go on here, and it was a very young field. So what did Barbara McClintock do as a crop geneticist? Well, she looked at what she could see under the microscope, namely, you guessed it, these chromosomes that we mentioned in the first part of the talk. Corn has 10 chromosomes, and what she would do is she would look under the microscope and see how changes in the chromosomes, whether they break in a certain place or whether something clearly flips around or joins with another chromosome, how that relates to what the corn actually looks like. And she would put it under the microscope like this, and she would see kind of all these weird patterns. Like you get some different coloration here, you get a normal one here. And what she found is that these differences in the coloration were linked to specific changes in the chromosomes in what they looked like under the microscope, certain breakpoints that seemed to move around the chromosome just kind of randomly. And that seemed to be linked to the shapes, the colors she was getting here. And we don't have time to get too into the detail today because it's a very complicated scientific story. But at the end of the day, what ended up happening is that what she was seeing were the very first transposons ever discovered. And for her work, it was published in 1950, and about I think 33 years later, in 1983, she was awarded the Nobel Prize in Physiology and Medicine for this contribution. And since then, there have been many, many transposons discovered, and they are incredibly diverse. And we can think of it almost as a zoo inside your own genome. And we're going to take this zoo analogy a little further. And if you think about an actual zoo, a lot of animals in the zoo, you have mammals, for instance, and you have carnivores, you have omnivores, you have herbivores, and you also have reptiles, for instance, some that slither, some that walk, and they're all incredibly diverse, but you can kind of fit them into families based on traits that are somewhat similar between them. And in much the same way, Milo already described TN5, which is a pretty simple DNA cut and paste transposon that has this one gene, this one protein that's encoded for by this middle blue bar. But as they started discovering more transposons, starting in the mid-70s, they found that this was definitely not the rule. You could have transposons that don't have repeats at the ends and that have totally different proteins that they code for. And you can have basically any variety of these that you can think of. And they're kind of associated into general families, just like carnivores and omnivores and walking and slithering ones. So the names are pretty cool for this. I have the year of discovery here so you can see what's going on, but you have Copia, Alu, L1, Gypsy, RTE, Penelope, Harbinger, Helitrons, Dashing, Krypton, Nagaro, Pollentons, and then all these other modified versions of these basic templates like Kolobach, Academ, Sola, Zisipton, and Zador. And these ones, the bold, were just discovered in the last 10 years. And so the idea of this slide, the key takeaway really is that these are kind of really diverse. They're they're replicating themselves in lots of different ways, and they're being discovered basically every year. You're going to find many new transposons discovered with the sequencing of new animals or other organisms' genomes. Not only that, but they're incredibly widespread across the tree of life. So if you check out the genomes of archaea, bacteria, protists, plants, fungi, humans, invertebrates, non-human vertebrates like fish, you're going to find that in basically every single genome you look in, there are going to be a number of these transposons. And they're 45% of Ryan Gosling's genome, of course. And they're a roughly similar percentage of the mouse genome. It's about 40% of their genome is made up of these jumping genes, these selfish replicators. But if you look at corn that Barbara McClintock was studying, an amazing 85% of that is transposons. But on the other end of the spectrum, you have this honeybee, the western honeybee, that only has 1% of its genome made up of transposons. So there's some weird stuff going on here that we still don't quite understand. So this is kind of amazing. But I want to just acknowledge that I've been pulling a little bit of a fast one on you guys, being a little coy, because this pie chart is true, but in reality, only less than 1% of the transposons in your genome are capable of actively jumping around. Just like you might have active volcanoes and volcanoes that aren't expected to erupt ever again in the history of time, you have active transposons that are capable of cutting and pasting or copying and pasting, and you have ones that, for whatever reason, can no longer do that. They can't make those proteins required for their own transposition. And the rest of those guys, we kind of call them a transposon graveyard. And they could be there, they could have been in the genome for hundreds of millions of years for all we know. And so to summarize this small segment, Barbara McClintock discovered transposons in about the year 1950. That's when she published her discovery, and she won the Nobel Prize for it in '83. Transposons are incredibly diverse, widespread branches of the tree of life, and abundant in most genomes. Yet, only a small portion of transposons in a genome will still be active. 45% of your genome is not jumping around right now. Maybe a very, very, very small percentage of it is. And the rest of it is kind of a transposon graveyard. And before we go on, are there any other questions? Yes?
When you give a 40 to 45%, is that typical of mammals, and is the 85% for corn typical of plants?
Right, so the question is whether the abundances I showed, the 40 to 45% or the 85% for plants, whether that's typical of the animals or plants respectively. I haven't looked at a big table yet to see if that holds true. Generally, what I know is that there can be tremendous variation in both those groups. So I think there are plants that have, as we saw, 85%. I think there are also plants that have even less than humans, less transposons than humans. And then other figures I've seen for animals include those like the honeybee, for instance, that are very few. But also, I think dog is about 41%. So it seems like that might be the average, at least. I think there are some fish that also have like 90%. Oh, yes?
We can make, what's the
That's a great question. So the question was, is this the maximum total number?
We can have like 20, we can't have 20 kids, we can only have like 5 kids. Like how much can they make?
Great question. So the question is, how many copies can a transposon make of itself? And it kind of depends. So a specific, let's say a cut and paste transposon, since it can't technically copy itself, it can only produce more copies in a way that probably produces one additional copy at a time. So it's just one kid per event, essentially. But a copy and paste transposon could have essentially unlimited number of kids. And that would happen not all in one burst, but it could happen over the course of many hundreds or thousands or millions of years.
Well, those are like grandkids and great-grandkids.
That as well, yeah. Yes?
I don't understand how you discover that this part of the genome is a transposon. What characteristic says that this is a transposon, so that you can say 45% of the total genome is a transposon?
That's a wonderful question. So for those who can hear in the back, the question is, what are kind of the telltale signs of a transposon? How can you, looking at a genome sequence, say, oh, look, there's a transposon? So for discovering new transposons, it's going to be a different process from finding a segment of the genome and saying that is a transposon that we already know and love. If it's a transposon that we already are familiar with, let me go back a little bit. As you can see on this slide, there are certain motifs that repeat themselves over and over again. So for instance, this piece right here, this is what's called reverse transcriptase. And this is in all of these, this type of transposon, retrotransposon, the LTR retrotransposon, and it's also in all of these, most of these non-LTR retrotransposons. And what that means is that if you can there's a program where you can type in a sequence, and if you type in the DNA letters for this piece, and you find it in different parts of the chromosome or in the genome generally, you can say with some reasonable confidence that, oh my gosh, that's probably a retrotransposon there. And there are other telltale signs as well, and people have very clever ways of doing this. Yes?
Are there transposons that have been observed as going from active to inactive?
Yes, there are, yes. And often, so the question is, have we observed transposons going from the active state to the inactive state? And one example of when they've seen this is they see a transposon, let's say this one. This is my favorite transposon personally, Taiwan copia. And what you can see is you can see it go to land inside the genome. And then it's just copy itself in there. But at that insertion event, there's some weird thing that happens where it gets kind of cut off, right? And so this piece to the left here gets lost and just goes spinning out into the rest of the cell and is degraded. And the only thing that's inserted is this segment. But this part alone isn't sufficient to jump around. And so basically, that whole sequence is now just kind of a dead transposon in there. And kind of interestingly, you can imagine that if for whatever reason the same thing happened right next to it, but it was only this piece, you could get the resurrection of that transposon.
Yeah, I was going to say, do they come back?
They can.
They go from active.
Yeah, it's a little bit like Pet Cemetery. They can come back. Yeah. Yes?
So based on this graphic, it seems like transposons can be different lengths. Is there like anything that kind of tells you how long it would be? Like what is the variance, I guess, in the lengths? Does that have any significance?
Thank you for asking that, because I've been meaning to make a point about that. So the question is whether what's the variation in the lengths of the transposon and the lengths of the DNA that is that transposon. And so to give two examples, the two two of the most common transposons, two of the best known. One is this guy, ALU. This one alone is about 10% of your genome. And this one is only about 300 letters long. Whereas this one, which is really abundant in the fly genome, in other organisms, including our genome, is about 6,000 bases long, letters long. And I think it can actually get up to, maybe 10,000 letters. But I'm not sure what the maximum they've seen is.
Thank you.
Yeah.
Does the amount of transposons in a living thing relate to the degree of that living thing to adapt the medium, to the ability of that living thing to adapt to the medium in which the living thing is living?
Yeah, thank you for that question. So the question is whether there's some correlation, there's some relationship between the number of transposons in an organism's genome and that organism's ability to adapt to its environment. And I don't know if anyone's actually asked that question before. If they have, I'm not familiar with the result. But as we're going to talk about later, Milo is going to talk about this specifically. There have been instances where transposons have been useful to the organism in adapting to a new environment. So how often it happens, I'm not sure, but it can happen.
Yeah, I think that's the kind of question that there would be scientists and researchers out there who would like to think about that question and might write down their ideas about it, but I think we don't know. But it's a really interesting thing to think about.
Great. All right, so we're going to move on to the next segment. Right, okay, so we went through the summary. All right, and so now we're going to start this showdown. And I'm going to try to convince you guys that transposons are parasites. They're out to get us. Because we already talked about how the genome is the instruction book for making you. Yet 45% of our genome are these active or dormant transposons. And to your question, sir, from what we can see, kind of on the face of things, transposons seem to serve no obvious purpose for your body's health and well-being. There's really nothing, as Milo said, they're selfish replicators. They don't care about you, they just care about making more copies of themselves. And so are they parasites or are they us in this more fundamental way? So I think transposons are parasites. Oh, and before I get to the next slide, a little trigger warning. There's going to be a gross picture on this slide. I kind of previewed it. It's really gross. But it's not like bloody or anything, but you might want to like cover your eyes quick or be ready to do that if you don't like it. And I'll tell you when we're off the slide. Thank you. So let's define what is a parasite. So a parasite, as I like to define it, and incidentally as Wikipedia likes to define it, go figure, is a relationship between two species where one, the parasite we say, benefits at the expense of the other, which we call the host. And so what we're seeing here kind of looks abstract, it's a little hard to make out, but here I think it's a human foot, and this is a little lesion, and it seems like there's some sort of string attached to that lesion and tied around a matchstick, and there's a little knot made in it. And what you're actually seeing is that this is a matchstick, and this is a guinea worm. It is really long. It's a parasite often found, I think, mostly in Africa. And what they've done is they've somehow managed to coax the worm out of the victim's body, the human's body, and they were able to tie it around the matchstick, and over the course of days, maybe even weeks or months, they very slowly turn the matchstick, or turn whatever rod it is, to pull it out of the body. And you really don't want to break it, because then I've heard very bad things happen to you if you break the parasite in you. And interestingly enough, just a fun aside, it's been suggested that this rod of Asclepius that's used on all these medical symbols bears a kind of maybe intentional familiarity between these resemblance to this process. So not sure if that's true, but I can see it being true. Okay, you can open your eyes again if you have them closed. So we often think of parasites in that mold, right? So there is something that's living inside you and feasting on your organs or whatever it is and doing really gross things. But parasitism kind of encompasses a much broader range of behaviors as well. For instance, here's a nest, This nest belongs to this guy, the reed warbler. So this warbler laid its eggs, but what's this egg all about? This is not a warbler egg. This is a cuckoo egg. And the cuckoo bird, what it does is it finds some unsuspecting mother's nest and lays an egg in it. And then what ends up happening is that this egg hatches, it kicks all the other kids out of the nest, and it basically just takes advantage of the mother. And this poor mother, for whatever reason, she can't recognize that this is not her own kin, and she feeds it and grows it to maturity, and then goodbye, says the cuckoo bird, and leaves this mother childless. So this is another form of parasitism where one organism, the parasite, is clearly benefiting at the expense of the other, the host. And yeah, I know, it's really sad. It looks like a good dude, right? Cuckoos. Anyway, so all of this is just to say that you can think very broadly about parasites. And we can ask the question, are transposons themselves parasites? And as Milo showed, they're selfish replicators. They're trying to make more and more copies of themselves. And by making copies of themselves, we can say benefit. And the question is, do they pose a cost for the host or the genome? And if we go back to our book analogy that we started with, imagine that somewhere in your genome, there's a beautiful line, Dickensian even. It was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness, it was the epic of belief, it was the epic of incredulity. The sentence that carries really deep meaning and is really important for the book, right? Now transposon comes to town. Bam! Transposon. What are you left with? It was the transposon best of times. Transposon was the transposon or worst of times. I can't even read it, right? So the meaning of this sentence is compromised by the insertion of these transposons. And in the same way, it's not hard to imagine how if this were, instead of a sentence, if it were a gene, and you had all these transposons landing in it, that could cause some problems for you. And we really don't have to look any further than the human genome and to human disease to see the signatures of this. And to your question, it was asked earlier if aberrations in organisms have been caused by transposons, and clearly there are examples where this has happened. There have been specific case studies reported in the literature where certain patients have hemophilia, lung cancer, colon cancer, and even other diseases that were the result of a transposon inserting itself in a very unfortunate place for its host. For instance, in a gene that's responsible for DNA repair. And so clearly, I think, I'm going to argue this, I think that these transposons are parasites where they are benefiting at the expense of their host. And now to go even further, kind of you think, okay, well that's not as bad as like viruses, right? Like viruses really screw up at the cell and they like move around and stuff. Well, I hate to tell you, but some viruses actually are transposons. So here's one of those LTR retrotransposons. If we put some color in it, this is kind of what it looks like where each color corresponds to a different genetic component. Now let's show like, oh, HIV, some retrovirus like that. Huh, it looks kind of similar, doesn't it, right? Basically, this is the same. And there's a little bit of difference here, right? There's some new stuff. But kind of the current state of the literature and the way scientists think about it is that actually what ended up happening somewhere deep in evolutionary time through one step or through many is that there was some LTR retrotransposon that somehow acquired some new swag, just like kind of inserted in there. And that swag allowed it to leave its own cell and then go on to infect other cells. But you know, it'd be one thing if transposons couldn't do this like now, but actually they kind of can, even if they aren't viruses themselves. They can actually hijack or be stowaways on other viruses to move to new organisms or new species. So for instance, there have been cases where this transposon here sitting in the genome is in the same cell as a virus. And that virus inserts its genome into the cell, and the transposon's like, oh, an opportunity. And it cuts or copies and pastes itself into the virus genome. And in so doing, when the virus actually says, okay, I'm out of here, it goes with it. And there's one case in particular that was discovered, I think, 5 years ago, of this transposon called SPIN. And they found this transposon in the genome of this guy. It's a blood-sucking insect, not from here, thank goodness, but from South America. and it was in the genome of Spin. And when they looked in the genomes of this cute gray short-tailed opossum, like, come on, right? When they looked in that genome, when they sequenced it, and when they looked in the genome of a squirrel monkey, they found essentially the same transposon in there. And they're like, what the heck is going on? Like, the same sequence, like 95% the same. And what they think might have happened is that sometime in the history of this blood-sucking insect that had the transposon, or something, maybe, like, I think it was more like this. A squirrel monkey had this transposon, and it got infected by a virus. The virus went into the blood-sucking insect. It was passed to the gray short-tailed opossum, and then it gave the transposon to the gray short-tailed opossum. So some weird story happened where these sorts of horizontal transfers are really possible. And to take this excerpt from this nice rhyme by Augustus de Morgan from 2 centuries ago, big fleas have little fleas upon their backs to bite them, and little fleas have lesser fleas, and so ad infinitum. Where the big fleas might be this guy, little fleas might be the virus, and the transposon might be even the smallest. So, to summarize, yes, transposons are parasites, I tell you. They reap benefits often at the expense of the hosts, including humans. Transposon insertions have been known to cause disease in humans, and what's more, retroviruses like HIV seem to have evolved from transposons. And even now, it seems like transposons can invade new species as stowaways on virus genomes. There's a lot more to go into here, but are there any questions? Yes?
Is there any way to stop transposons from like going into like a virus or like another kind of chromosome?
So the question is, are there ways to stop transposons from going into viruses or other parts of the chromosome? And the answer is, there certainly are ways. If you knew at least a specific transposon you wanted to stop, there are things you could do. But for the most part, to actually put these into practice, say in a human, it would be really hard to do that kind of as doctors or something like that. But as Milo is going to discuss, there are things that have been done by your body itself over evolutionary time to stop transposons from moving around so much.
We're going to do 2 more questions. Just
Great. Yes?
In the National History Museum in Harvard, there is an exposition of evolution and how in islands like Hispaniola or Madagascar, where there's different species, they go very different than in land like Africa or places. So is any kind of environment when the species are more susceptible to have this kind of situations, like you can see in those islands, Madagascar and Hispaniola?
That's a good question.
Like, let's say, when it's very cold, maybe the transposons are not very active. When it's warm, it's more, or when it's humid, you know?
Yeah, thank you for that question. So the question is whether kind of transposon activity could be associated with not just specific environments, but even kind of over evolutionary time with certain geographically isolated places. And the answer is, at least to the activity part, is very much yes. And for instance, one of my favorite transposons that infects yeast tends to be more active, kind of to wake up when the temperature is really low. For whatever reason, it does that. And I don't think it's quite understood why. So certainly transposons themselves are changing what they do in response to bigger environmental factors. All right, one more question. Yes?
That process you mentioned earlier about taking the human being able to splice into the genome what we want and put it in a particular place.
Yes.
Can we do that with transposons?
Yes, and actually, Milo is going to talk about a similar process that scientists are using with transposons to make those sorts of insertions at the end of the talk. Great. So I think we're going to move on. Right.
So before the break, Chris kind of made this argument that transposons are parasites. And not to take away from the extreme drama of the situation, but I'm not going to specifically try to convince you that transposons aren't parasites. But I'm going to try to convince you that as parasites, they're kind of what I'm going to call considerate parasites. And And in a lot of other ways, transposons are really us. So the first point is that transposons take up a huge portion of our genome, so we have some intuition that they kind of are a part of us. They're in our genome. So in trying to convince you that they're considerate parasites, I want to talk about different parasite strategies. And I think they fall along kind of a spectrum from fast and aggressive to slow and low impact. And so in thinking about these kind of strategies for parasites or for other kinds of threats or diseases, we can think about this spectrum. So up in the fast and aggressive corner, we might have these little viruses called bacteriophage. And certain bacteriophages, when they attack a bacteria cell, they'll go in, make tons of copies of themselves, completely destroy the cell, basically, and the cell will actually explode and let them out, and then there will be tons of new copies of these bacteriophage. So this is kind of the most aggressive you could be. They get in, they get what they need to do done, they get out, they don't care that they've just totally destroyed the cell that they were living in for a second. Somewhere in the middle is kind of how oftentimes we like to think about like the common cold. So if I were to catch the common cold and then just immediately die, it would be hard for the common cold to find a new host, right? So by like being a little bit low key and letting me live a little bit, I might sneeze on Chris and get him sick. And so in this way we can think of parasites or these threats kind of having to balance being fast and aggressive versus being able to live in their host for a while and get passed on to a new host. So I'm going to argue that transposons are on the kind of far end of this scale, really slow, low-impact parasites. And just to give you a metaphor for this, if we think of parasites as house guests, I think the virus is kind of like this guy that says, hey man, I'm going to come crash at your house with my 20 friends from college just for the night, see you soon. And they're going to come in and they're just going to cause a disaster in your house and they're going to have a good time and then they're going to be gone the next morning and they're going to go on to the next thing and they don't really care that they destroyed your place. And I think transposons, on the other hand, are more like these little mice that are kind of living in the corner of your house. Maybe you hardly ever see them. They don't cause you many problems. Things like that. So why might we think that transposons are going to be like this, are going to be considerate parasites? So I think the kind of basic answer is that transposons are really committed to this cell in a way that other parasites aren't. And they're committed to the cell because they put themselves in the genome. You can't get much more committed than putting yourself in the genome of a cell. So if we think of a transposon's goal as being to make a new transposon to cut or copy and paste itself, first it has to stay alive. So if the cell that you copy and paste yourself into immediately dies, you as the transposon are also going to die, and that's not going to be good. So in this way, the fate of the transposon and the fate of the cell is kind of aligned. Their fates are intertwined, and they in some senses have the same interests. So this might lead us to suspect that transposons might actually do some things that are kind of considerate and kind of nice for the cells. So what are some examples of this? First of all, and there was a question earlier, I can't remember from where, but there was a question earlier about whether transposons insert themselves randomly in the genome or insert into specific places. And so the point is that where you paste yourself really matters, as we said before. If you paste yourself in the middle of an important gene that's going to cause problems, the cell might die and you won't get passed on. So Chris showed this, this neat little metaphor where the transposons have inserted into this passage, but I just wanted to point out that we were actually kind of nice here. And if we really inserted them randomly, it would be even harder to read, and it could be much, much worse. So in the previous example, we had transposons always landing between words, and it actually isn't that hard to go through and read it. But if they really insert randomly, it could be much worse. So this is one of the ways that transposons are kind of considerate house guests. We know of lots of transposons that exhibit what we call site specificity. So if you're a transposon and you evolve a way to, instead of every once in a while landing in an important gene where you cause the cell to die and you cause yourself to die, if you can evolve a way to try to land instead always in front of that gene, for example, then that can be beneficial for you, beneficial for the cells, beneficial for everybody. So this is something that, again, it's different between different transposons in that zoo of transposons, but for many of them, they've evolved different ways of being able to specifically avoid inserting into regions that will cause problems. A second example is that transposon proteins, if you ask someone who studies proteins in cells and they look at how these molecular machines work, they'll tell you that a lot of these transposon proteins are actually really low activity. And at the end of the talk, when we talk about using transposons for things that we want to do as scientists, we actually have to try to kind of up their activity because they're so slow. And this may be another way that transposons are kind of being considerate houseguests. You can imagine if you go into a genome and you're quickly copying and pasting yourself all over the place, you're going to cause lots of problems. And again, because your fates is intertwined with the cell, that might be bad for you. It's a little tough, again, here to disentangle exactly what's going on, because an alternate thing that could be happening with low activity is that the cells could be evolving ways of preventing the transposon protein from being as active as it wants to be. So it's a complicated kind of give and take, but we've definitely observed some of these things that seem to show the transposon acting in the interest not just of itself, but kind of the cells that it's in as well. So the next example is slightly different, and it's a specific way that our cells in our body have figured out how to, what I'm going to call domesticate transposons. And so this, I include this picture for this section not because the domestication of dogs by humans is a particularly good metaphor for the domestication of transposons by the body, but because this is my friend Francis's dog Noli, and it's a very cute dog. Okay, so I'm going to have to go on a little bit of a tangent into some biology, but I promise I'm going to get back to the way in which the body has domesticated transposons to use in this system the adaptive immune system. So the adaptive immune system is a fascinating thing, and there could be a whole Science in the News talk on it, which I'd really like to see. But today I'm going to kind of give you a cartoon version of it and a cartoon version of a specific problem that our immune system faces. So the problem is that we see lots of threats from outside. These could be bacteria, these could be viruses, things that pose problems to our body. And one of the tasks that our immune system has is to recognize the threats. And so to recognize these threats, one thing that our immune system does is it produces these proteins called antibodies. And we can think of an antibody that recognizes a threat as kind of being a lock and key mechanism. So if you have an antibody that matches a threat, it can recognize that threat. And if you have an antibody that matches a threat, your body can also kind of remember that it saw this threat, make more of this antibody, and you'll be better prepared for that threat next time you see it. So the problem is, we can imagine if there were only 2 threats in the world, then we could just have the 2 antibodies we need and it would be no problem. But the problem is there's many more threats of all different kinds of shapes, and in order to make all the antibodies that we need to recognize all the potential threats, we need to have just an enormous number of proteins, an enormous number of genes to make those proteins. So the way the body has solved this problem is to break up these antibodies into these kind of modules. So you see I have them colored here in 3 different colors, because these are kind of 3 different modules that are hooked together. And what it can do is it can make combinations of different blues, greens, and yellows. So I like to call this the antibody slot machine. And the 3 segments are called V, D, and J for reasons that I don't think are important, but I kind of like the letters. So you can think of it just like a slot machine. There's many versions of V, many versions of D, many versions of J. So in order to make a bunch of different antibodies, the body will take a spin on this slot machine and end up with one combination. And then in another cell, it'll take another spin, and you can end up with a completely different combination. So with just something like 40 V's, 30 D's, and 6 or 7 J's, you can end up with something like 8,000 combinations. And then with, in reality, there's actually one antibody with 3 segments and another with 2. And altogether, you can end up with over 1 million different antibodies just from making combinations of less than 100 things. So you end up with these different combinations, and if one of these recognizes a threat, then the body can remember that, and next time you'll be ready for it. So the question here, bringing it finally back to transposons, is how does this slot machine work? And it turns out that some of the proteins that are involved in this that scientists have found are called RAGs, recombination activating genes, and it turns out that they're actually transposon genes. So kind of in a similar way to how Chris described these viruses transitioning from being transposons to being viruses, it looks like these genes have transitioned from being transposon genes to being these RAGs that are used by the body for this process. So what the RAGs are actually involved in doing is making these connections between V segments and D segments and D segments and J segments. So I'm going to show briefly what this looks like because you'll get an idea of why it's similar to transposons. So the way this actually works is that you have a set of V's, and this is DNA now, so the V's are in the genome and they look like this. And I have 2 V's here and 2 D's because I'm showing just the simplest example, and I'm just going to focus on the problem of how you have a V and a D and you match them up and you make different combinations. In reality, there would be like 50 V's, 30 D's, and some J's somewhere over there. So each of these V's will code for kind of a different structure protein that can be matched up with a D and with a J. And so I've showed here under what my little cartoon protein shapes would be for each of these DNA segments. So you'll notice that I put these kind of familiar brown edges next to each of them, and that's because there's a certain repetitive little sequence here, which is, for our purposes, similar to the edge of a transposon. So what the RAG is going to do, just in the same way that we've seen TN5 work, is it's going to cut out, just like a transposon, between 2 of these brown segments. So as a first example, it might cut out just those 2 brown segments there and pick up that little section like a handbag. In this case, the handbag is just trash. It's just going to get thrown away. But these 2 segments that have been left now are now available to come together and meet up. And now when you make a protein out of this section, you'll have the V2 attached to the D1, and you'll get this combination. So just to show that you can make different combinations, if instead it cuts between this brown segment and this brown segment, you'll pick up with D1 in it this little handbag. Again, that's trash, and this time V2 and D2 can come together. And this process of them coming together is complicated and involves other proteins. But the point is that the RAG, by cutting things out, allows them to come together. And so you can get this other combination. So obviously here I've shown kind of the simplest possible example of just 2 V's and 2 D's, and you could imagine the 4 combinations you can make. But if you had 50 and 30 and 7, you'd be able to make many more combinations. And so it's really a cool example of how over evolutionary time, at some point, the body managed to take a transposon gene and start using it for its own purposes. And importantly, this RAG gene does not do any normal transposon stuff. It's not going to copy and paste anything to other places in the genome. It's just working for the body at this point, and this is all it does. So it's kind of the special example of a way in which the body has taken transposons to use it for its own good. So in summary, while transposons might be parasites, they also live inside our DNA. And in many ways, our fates are intertwined. So this has led to many transposon behaviors that reduce impact on the cell. And finally, the cell has even domesticated transposon technology for its own use. And so now I can stop and take questions.
Yeah. So you were just saying that the transposon mechanism is used for in this immune response. Do we know which came first? Whether transposons are sort of a corruption of the immune response, or vice versa?
It seems, so the question is which came first, this kind of immune system protein or transposons? And it's clear from the evidence, if you compare the sequences, that this immune system protein came from transposons. So transposons came first. But at the same time, you might wonder, where did transposons originally come from? And it's certainly possible that there's some sort of similar system out there that became transposons in kind of the reverse way. But it's not this system.
A general question about transposons. It seems like the difference between a transposon and a gene is the ability for it to cut itself out of the sequence and then move somewhere else to create a protein. Is there like a fundamental part that's similar between all transposons that performs the cutting function, that moves around different bases?
So the question is whether the part of the transposon that does the cutting or the copying and pasting is some fundamental part that that all the transposons use. And I think it's difficult to answer that question, certainly for DNA transposons, because they're so widespread across the tree of life, and so there's a lot of proteins that are similar, but there are also ones that seem completely different. It seems like there are certainly some that are completely different. I think the closest thing to what you're talking about is probably what Chris mentioned earlier for copy and paste retrotransposons. The reverse transcriptase is a certain protein that they use to do that, and pretty much all of them use that protein.
Do you mind if I make one more minor point? We didn't include this in the formal presentation because we didn't have time, but as a rule, all transposons are able to copy them or cut themselves out and paste themselves somewhere else. But there are actually some transposons that have parasitized other transposons. We say, all this copying and cutting machinery, I don't need that. I just need the recognition sequences. And by having those recognition sequences, the proteins that other transposons make will take me up like a handbag and put me somewhere else. And so there's actually a really cool dynamic there as well. Yeah, that's all.
One more question.
Yeah.
Do you notice that transposons may be located in different places in different somatic cells of the same organism? For example, that they might be on different chromosomes or be more numerous in some somatic cells rather than others.
Yeah, so the question is whether transposons are, well, let me see if I understand the question right. So the question is whether transposons are in different places in different...
Of the same organism.
In different parts of the same organism.
So that
Yep.
over time, as the organism is developing, different somatic cells may evolve transposons in different ways.
Yeah, absolutely. So, just for people who don't know, the somatic cells are kind of like all the cells of you that aren't going to be passed on if you have kids.
For example, would liver cells potentially be different from skin cells in this regard?
Yeah, so I don't know about like a specific point about whether liver and skin would be different, but certainly this is something that happens. And with Chris's examples of, for example, cancers, this is exactly what's happening. A transposition event has happened. It's changed some cells, and those cells have become cancerous. And almost certainly it's happening without major effects in different parts of the body all the time. And there are some transposons, just again to make my point that they're kind of considerate house guests, there are some transposons, like the P element, that have evolved ways to not do that, to only transpose when they're in the germline, when they're in the cells that are going to be passed on to the next generation. And so by only doing that, they're kind of being nice to the rest of the animal. They're not going to cause these problems. Great. So with that, I'll head on to the next section, which is engineering with transposons. And so in this section, I'm just going to tell you kind of a few fun examples of how we're using transposons in the lab to do our work for us, basically. So the genome is this big book that we care a lot about for all these reasons we've been talking about. It'd be great to know how different systems work, how humans work, how microbes work, all these things. And there's a few things specifically that we'd like to do with the genome. We'd like to read it, and we'd like to be able to edit it, to be able to kind of probe how it works. So transposons, it turns out, can help us do both of these things. So how do we read genomes? Well, there's a lot of ways to read the letters in your genomes, but one of them that's really popular right now, one of the technologies out there, is Illumina sequencing. So this is a really professional scientist next to a machine called the NextSeq. And this is a machine that can read DNA. So if you put molecules of DNA onto this machine, it can read the letters off of the molecules, and you can get huge files to fill up your computer with tons and tons of letters on them. So importantly, the way this machine works, it reads short pieces of DNA. It can only read about 300 letters at a time. Now, luckily, it can read like 100 million stretches of 300 letters, but it can only read 300 letters at a time. So that might seem weird in terms of being able to read a genome that's really long. You'd like to be able to read more than 300 letters. And there's other technologies out there for that. But with this one, the way you get around it is that if you know kind of what the genome looks like, you can take your short little 300-letter read, and you can line it up to your genome. And if you get enough of these, you can line them all up, and you can kind of piece together what you're seeing. So this is the way this machine works. So in order to use it, we need a couple things. We need short pieces of DNA, which I'm showing here as these little gray blocks. We need to have short pieces of DNA. And then the other thing we need is we need to have special little pieces of DNA on the edge, which I'm calling adapters. And the reason we have these little special pieces on the edge is that, one, the pieces of DNA have to attach to this machine. And so you can think of this kind of like these are little pieces of Velcro, so that when we put the molecules into the machine, they're going to stick to it, and we're going to be able to read them. And the second reason is that we need kind of a place to start reading from. So since we have these little adapters and we know what they are, we can start reading from there, and we can read our 300 bases. So we have two tasks as biologists if we want to do this. We have to break up the genome into little pieces, and then we have to attach little pieces that we know, these adapters, to the side. And it turns out that we know something that's really good at making cuts and attaching pieces of DNA, and that's a transposon. So TN5 actually has been engineered to do this. So just as a reminder, the way TN5 works, it's carrying around this transposon. It makes a cut in the genome, and it unfurls it in there and sticks it in. So what scientists have done is they've engineered what I'm going to call super TN5P, not the actual name, but I like it. And this is a hyperactive version of TN5P. So it's going to work better. This is going back to the low activity point.
Real quick, TN5P
Yeah.
is a transposon?
So TN5P is a transposon protein. So transposon we would consider the piece of DNA, and it makes TN5P. But in this situation, we're just working with it. We don't even need to think about that. We just have the protein. I buy it online. Amazon Prime. So we have super TN5P. It's a hyperactive protein. And what they've actually done also is they've attached these adapters to it. And these adapters kind of look like the ends of transposons. So they fit right up in there just like the ends of the handbag do. But this time we've kind of tricked it. There's no handbag. There's just these two little runt ends sticking out. So this is the system that we make in order to do this. And then we put TN5P to work on our genome. And it will go make a cut. And it will try to insert this fake transposon. But it's just going to attach these little blunt ends. And it'll leave a space in the middle. So we do this many times. all over the genome. We just put in lots of this protein along with the adapters. And you'll see, just like that, we end up with exactly what we wanted. We end up with a lot of small pieces of DNA with adapters attached to the end of it. So this isn't the only way that you could get this done. I don't want to convince you that like we couldn't do the sequencing without the transposon protein. But it does make it much easier. It kind of takes 2 hard steps and makes it 1 easy step. So that's one way that we've figured out how to use transposons in the lab to actually understand what our genomes are like and what the genomes of microbes are like. So this next example is something that I'm actually working on right now, and it's studying the genome by breaking it, and it's called transposon mutagenesis. So if we think about my favorite organism, yeast, which are the little microbes that help you make bread and beer and wine and these sorts of good things, and we think of them as having this single chromosome genome. They actually have several chromosomes, but we'll just think of it as 1 for now. We can think about putting transposons into yeast artificially and having them insert. And we can have them insert in random different places in the genome. And this is something we can do. So this is a way to cause mutations in our yeast. So why would we want to do that? So just to take a totally hypothetical example, we might be interested in how yeast can grow at different temperatures. So we know yeast likes to grow at, say, 86 degrees, but we also know it can kind of grow okay at 100 degrees in really hot environments. And we'd like to know, why can it do that? What is involved? We just want to know more about the biology there. So one method we can use with transposon mutagenesis is we just break everything, right? We're just going to take a ton of yeast, and we're going to do this thing where we put the transposons in and we have them do insertions, and we're going to end up with tons and tons of insertions in tons and tons of yeast, so that we're really breaking in single cells everything in the genome. So then we can kind of do this simple experiment and look for effects. So for a certain yeast line that has a certain insertion, for a certain type of yeast that has a certain insertion, we can measure how fast it can grow at 86 degrees and at 100 degrees. And if this insertion is just in a region of the genome that really doesn't matter and isn't doing anything, maybe it just grows the same as it would have with no insertion at both temperatures. But if this insertion is in some region of the genome that's actually really important for growing at high temperature specifically, we might see this pattern where it grows fine at normal temperatures with the insertion, but at high temperatures with the insertion, its growth rate is way decreased. And you might see crazy interesting things like, okay, this insertion caused it to grow worse at low temperature, but now it's growing even better at high temperature. And so by doing this, you kind of are taking a shotgun approach of just trying everything, and you're getting some possibilities for genes that could be involved, because we can go in and we can see where this insertion is. And if this insertion is in a specific gene, we might start to think that gene might be involved in growth at high temperature. So again, it's kind of this shotgun approach, and it will give us genes that we should look into further if we're really interested in this. So in summary, transposons are parasites, but they're parasites that have very specific skills. And these are skills that we can use to read the genome and skills that we can use to make these large sets of mutations in microbes, which can allow us to identify important genes. I can stop if people have questions about this engineering stuff.
Was the transposon sequencing, is that shotgun sequencing?
I'm using the term shotgun like too freely. The transposon sequencing is I don't know, it's Illumina sequencing.
Yeah, it's an example of shotgun sequencing.
It is shotgun sequencing?
Yeah.
Okay. Got it. These guys know.
Only time for one more question.
All right. If not, we'll have time to talk after the talk if people are still interested. So I just want to go over what we talked about today. So first of all, we went over kind of what transposons are and how they work. And then we talked about their diversity and their abundance and how they're widespread across the tree of life, as well as the story of Barbara McClintock discovering them. And then Chris and I kind of talked about, are they friends or enemies? Are they parasites or are they us? And then finally, I was just talking about kind of ways in which we can use transposons to do work in the lab. So in conclusion, I think I'd like to say that yes, transposons are parasites, but at the same time, our fates are intertwined in a lot of ways between the host and the transposon, and so they're also us. And then finally, that transposon technologies are providing these ways to help us better understand transposons and ourselves. And so with that, we'd like to thank Steph and Katie, who've been helping us out a ton with this, as well as the fall series directors, Andy, Heather, and Kayla, and the group that will be presenting next week, Christian, John Mark, and Michael, for giving us feedback when we did our practice talk. Everybody's been really helpful. And we should also thank all these great organizations that provide support for this kind of fun lecture series. And thank all of you for coming and listening to us talk about transposons for a long time.